After explaining how glucagon activates glycogenolysis, now we will talk about the process of activating glycogenolysis through epinephrine. So epinephrine has adrenergic receptors on the surface of hepatocytes, alpha-adrenergic receptors and beta-adrenergic receptors. Once epinephrine binds on beta-adrenergic receptors, it will act exactly like a glucagon, so we're not going to re-explain the steps. However, when epinephrine binds on alpha-adrenergic receptors, it will activate glycogenolysis, but in a different way than that exerted by glucagon. Same as that for glucagon receptors, epinephrine adrenergic alpha receptors contain a coupled G protein with alpha, beta, and delta, with a GDP molecule, of course. Once epinephrine binds on alpha-adrenergic receptors, this receptor is going to be activated by the conversion of a GDP into a GTP molecule. When GTP molecule is present, the alpha subunit will dissociate from the delta and beta, forming PPI2 molecule. This PPI2 molecule is the release of 2-phosphate group from the phospholipid bilayer through the activation of alpha subunit by a GTP. These two inorganic phosphate groups will be converted into IP3 and DAG by the enzyme phospholipase C. So this phospholipase C will convert the two inorganic phosphate groups into isotyl triphosphate and diacyl glucose. These two molecules, the DAG and IP3, both considered a second messenger, but each will act on his own way for the activation of a glycogenolysis. Let's start with IP3. This IP3 will work on the calcium channels of phospholipid bilayer by activating them, allowing the release of calcium inside the hepatocytes. This calcium released will bind to calmodulin, a compound forming calcium calmodulin complex, where this formation, the calcium calmodulin complex, will form a calcium calmodulin dependent protein kinase. So that means the formation of calcium calmodulin complex allow the activation and the functioning of a protein kinase. Once this protein kinase is active in the presence of calcium calmodulin complex, by the use of an ATP molecule and energy molecule, it will perform a series of phosphorylations, allowing the activation of glycogenolysis. However, on the other side, DAG, the diacylglycerol, will activate protein kinase C. Once this protein kinase C is active, it will use an ATP molecule, an energy molecule, also performing a series of protein phosphorylation, activating the glycogenolysis. Keep in mind that this phospholipase C is the same phospholipase C inactivated in the pathway of activating glycogenolysis by protein kinase A. So this phospholipase C inactivated by inhibitor 1 is the same one used for activating glycogenolysis through epinephrine. Also, don't forget that the phosphorylations occurred in the activation of glycogenolysis through glucagon and through epinephrine on the alpha-adrenergic receptors occurred on threonine and serine residues. And by this, we will end the topic. Hope I make it clear for you. Don't forget to subscribe, and I will leave links in the description for other YouTube videos. Thanks a lot. After explaining how glucagon activates